Hello everyone, my name is Frankie Foltz, and it is my pleasure to introduce you to the research I've been working on for the better part of my undergraduate career, investigating the non-invasive methods of hormone quantification in great black-backed gulls. But wow, that title's a mouthful, so let's go ahead and break it down. Investigating. This is an ongoing process of establishing methods for the overall goal, which is non-invasive hormone quantification. We want to know the testosterone levels of our focal individuals who happen to be great black-backed gulls. In the next 15 or so minutes, this is what we'll be going over. First, a bit on the field that studies the intersection of endocrinology and behavior, then a bit on gulls and specifically our study species of great black-backed gulls, the overall study design including the fieldwork, and finally, my role in the project, developing methods, determining the validity of these methods, and what we're working on now and in the near future. So the link between testosterone and aggression has been established or refuted for many mammalian, reptilian, and avian species. On this side, I put just a few of these studies done on avian species. But there's something peculiar here, and that's that all of these have been done on passerine species. And this is great. We love passerines. They're super cute, incredibly abundant, and integral to ecosystems everywhere. But if you just add the word goal to your search, you suddenly go from 241 results to 43, most of which really shouldn't be there. But anyway, all this is to say that there are many differences in passerine species and non-passerine species, but there's a huge study bias that provides more information on songbirds than any other order. As this phylogenetic tree shows, though, there are many other orders that need to be studied as well, including the R order of interest that includes shorebirds, also known as Charadriiformes. And I apologize to any of you if I've said that incorrectly. The internet seems very divided on pronunciation. So why did we choose the Gulls of order Charadriiformes instead? Well, if we're looking at physiological causes of aggression, we are interested in a species that is known for its somewhat legendary aggression. Enter the great black-backed gull, a species that, if you want to study, you are legally required to wear bike helmets in the field because, as you can see, our wonderful field workers are about to get absolutely dive bombed. There's one thing that gulls have that other groups just don't: the audacity. And if you're anything like me, you'll witness this phenomenon of getting wrecked by a 4-pound bird, and you're probably wondering, where do they get the audacity? And the most likely culprit for that is testosterone. As many of you likely know, testosterone is often misrepresented as a male hormone that's found in most vertebrates. As you can see from this diagram, it's a bit narrow-sighted to pin it only as a sexual hormone when it has strong implications for several biological functions. Like many biological molecules, it's a double-edged sword. So over here on the right, we have the effects of testosterone that should theoretically aid in the individual's lifetime reproductive success. But on the left, we can see all the negatives of testosterone. Thus, the hormone exists in a threshold that allow for optimal benefit with minimal side effects, which leads us to our question. Does testosterone mediate aggression in colonially breeding great black-backed gulls? And if so, what range of values correspond with more aggression for both males and females? Here you can see just a few examples of this aggression towards others. Counterclockwise from left, we have this individual likely defending territory, this juvenile attacking an adult herring gull, this individual nest guarding, these two doing a long call, likely as a territorial warning, an adult putting the juvenile back in its place, and finally up here, a great black-backed eating another of its kind, likely after a territorial dispute. So how do we measure these testosterone levels, though? The traditional method is through the use of plasma or blood samples, and this makes perfect sense. As we can see from this simplified diagram here, hormones are manufactured in a source cell, then secreted into the blood stream where they will travel until reaching a target cell, which is just any cell that has a specific receptor for that hormone. Blood sampling takes the hormone straight from the blood, capturing the exact concentration of hormones circulating at the time of the blood draw. It's important to note, though, that while plasma sampling is the gold standard, it still is fallible. Yes, you do have a reading of how much hormone they had at the time of sampling, but these only tell you about the time of sampling. For instance, take these two hypothetical graphs of hormone measured from two different individuals. Both had plasma readings at the same levels at the same time. However, what happened around the time of sampling was a completely different story. Bird 1 had a more gradual climb and sustained higher levels, plus a second time where its levels peaked. Contrarily, Bird 2 only had one quick spike before returning to its baseline. From only plasma readings, these two would look exactly the same on paper, even though they experienced very different situations. There is a method that would allow us to distinguish these differences, though, and this is fecal hormone metabolite sampling. Hormone metabolites are the used processed hormones that accumulate over a range of time, which is variable for different classes of animals, and then are excreted out. The concentration of hormone metabolite in a fecal sample, then, is representative of the integral hormone concentration over a certain amount of time. In other words, the plasma samples would give you these red lines, which are identical between the individuals, and the fecal samples would give you the entire shaded regions, showing that bird 1 has higher levels throughout the period than bird 2. To summarize, here's a quick comparison of the two methods. As we just discussed, plasma sampling reflects the hormone signal at a single point in time, where fecal sample reflects the hormone signal over an extended period. Another seemingly small but important distinction to make is that plasma sampling measures hormones, where fecal sampling measures hormone metabolites. After long last, we get to the non-invasive part of the title. Plasma sampling requires the capture and manhandling of focal individuals, but fecal sampling only requires a field researcher to collect an individual's fecal sample after defecating. This has many benefits, including decreasing the risk of artificial spikes from the stress of capture, as well as decreasing the risk of injury both to the animal and the field worker. And finally, with the hazards inherent to plasma sampling, it's not really ideal for repeated sampling. However, repeated sampling is definitely possible for fecal sampling because it can increase the accuracy, not to mention it doesn't really affect the birds. How do we go about doing this? First, a field worker, note those helmets that we pointed out earlier, approaches a nesting gull and will stand 1 meter away. No antagonizing, no yelling, no waving your arms like a crazy monster, no nothing. Just standing there. The field researcher will then try to stand in that spot for a whopping 30 seconds and record the gull's response on a scale from 1 to 8, with 1 being no response and 8 being a direct attack. It doesn't really sound so bad, right? In case you're wondering what this would look like, here's a quick clip that my supervising professor took just while walking past the nesting pair. You see they start to approach, a couple calls, a bit of a charge, some more calls, another charge, and then we have a flight and direct attack from the second one. Now, interestingly, that happened when they were 3 meters away or so and just walking by. Can you imagine just standing there? But anyway, after the aggression test, the field researcher will try to coax the gull onto our very carefully constructed poo mats, where we hope they will drop a deuce. While this happens, a researcher is observing so that they can match the bird to the fecal sample using their tag. I zoomed in on this picture so you can hopefully catch a glimpse of this tag on the gull's leg. But anyway, this fecal sample is then scooped into a centrifuge tube for storage, quickly vortexed, and put in the -80 freezer. After all the samples for the season have been collected at Appledore, which is up here on the left, they are shipped back to our home base in Los Angeles at Loyola Marymount University. Here I, along with my fantastic research partners Allie Waller and Katie Inkrot, process these samples to determine the testosterone metabolite content. But anyway, sounds pretty straightforward, right? So here's an overview of the lab methods and how we thought they'd go. Freeze dry samples, conduct extractions, then run enzyme immunoassays. I can't remember the last time anything in life actually went as planned though, so in the next slides, I'm going to tell you how things actually went. So let's break it down. Here's the first step in some of what we encountered. The first step to establish was freeze drying using this lovely contraption up here. Luckily, LMU already had a lyophilizer, but no one had used it in years, so we got the formative experiences of reading equipment manuals and figuring out how to change the oil on a vacuum pump. When it came to actually freeze drying the samples, we checked their weights every 4 to 6 hours, including overnight. We found that for the most part, it took less than 12 hours to completely dry samples, as illustrated in this graph. Also fun with the freeze dryer was that sometimes when you hooked up a sample to the vacuum, it would shoot out of its container and just get all over the flask, and we'd lose a whole sample that way. To avoid these poop explosions, as we called them, we devised a very high-tech method of putting 2 layers of Kim wipe taped over the top of the microfuge tubes that the sample was being dried in. We also determined that this was due to the samples not being entirely frozen through, so we figured out to take the samples out and prep them, then stick them back in the -80 freezer for a while and only take them out directly before hooking up to the freeze dryer. With this modified method, I can proudly say that it has been 572 days since our last poop explosion. Woohoo! Up next was the process of determining optimal grinding and extraction procedures. When we were working with the live fly samples, both in trying to grind them and in weighing them out, we found that the powder was so light that it was liable to just fly out of the tube of its own volition. To prevent this, after extensive searching and trial and error, we were able to get a plastic draft shield that is supposed to be used at a bakery, but hey, if it works, it works. With this protection, we were able to successfully weigh out our 0.1 gram aliquots of powdered samples for the extractions, as in our literature search, we found that that was the most commonly used, in an extraction ratio of 0.1 grams of dried fecal matter to 2 milliliters of solvent. Also from our literature review, we were able to devise this scheme for extractions. Testosterone was extracted with 80% methanol, then set to shake on a vortex for 30 minutes before being centrifuged for 20 minutes. I'd also like to point out that we did, after a while, realize that there was an adapter for a vortex that we could use instead of this janky setup. 0.5 milliliters of the extract after centrifuging was saved for the single extract, while the rest was set aside to be combined for the second extract. Extraction procedures were then repeated, and the supernatant was added to the 1.5 milliliters that was set aside for a total of about 3.5 milliliters. So this allowed us to, once we ran assays, compare how much testosterone we extracted with each round and if it would level off after a certain number of extractions. But why not just repeat this several times, up to 5 or 6? The answer for that lies in evaporation times. Most enzyme immunoassays will bug out and not work accurately if your sample contains alcohol. So we tried a variety of methods to get the alcohol to evaporate off and leave the testosterone behind. Other papers suggested leaving samples out overnight, but that barely took the levels down. Heat blocks wouldn't effectively heat the conical structure of the microfuge tube, and a heat vacuum setup greatly increased the risk of flash boiling. What we settled on then was a hot water bath and using nitrogen for the last 30 minutes or so of what would be a roughly 12 to 24-hour procedure. All this is to say that with more rounds of extraction, we have more liquid that needs to evaporate off, and that means more than a day in a hot water bath. Anyway, we were all set to run these assays and compare the single extractions to the doubles, at which point we encountered a nearly 9-month delay due to the pandemic. It was only this semester that we could actually get data to work with. When we were finally able to get the assays completed, we found a wide range of values. Per milligram of fecal matter, there was anywhere from 8 to 226 picograms of testosterone present. The maximum was over 30 times that of the minimum. This histogram shows the distribution of these values. As of now, there's a right skew, but that makes a bit of sense. You can't really go below 0 when measuring these values, so we really only see the tapering on the right side. But why is there so much variation? A lot of this has to do with the fact that these individuals are likely all at different stages in their reproductive cycles. Butler and James Butler found that great blackback gulls are in fact more aggressive after their chicks have hatched. Therefore, if testosterone and aggression are linked, we would expect that testosterone levels would be higher along with those aggression levels after the individual chicks have hatched. In this chart, we have a box plot comparing the testosterone value, again in picograms of testosterone per milligram of fecal matter of single extracts to double extracts. A paired t-test between single and double extract returned a p-value of less than 0.01, which is below our alpha cutoff of 0.05 and therefore means there is a significant difference between the two groups. But what does that actually mean? The double extract return on average a concentration of testosterone that is 176% more than that of their corresponding single extracts, not really a negligible amount. With this in mind, we are now questioning if the double extracts are getting as much as they can, and we are currently in the process of testing triple extracts as well. We may even go up to quadruple. I want to point out that we still have 2 outliers, one for each group, and these come from the same individual. Something that we'll be looking more into in the near future is comparing these fecal readings to the plasma samples that we took and taking another look at this individual to see if they're still high above the rest of the group with the plasma samples. And then in the future too, the field researchers will be recording those hatch dates for each nest, which is linked to an individual. And then we'll be able to compare testosterone and aggression along a timeline relative to hatch dates. Even though we should never fully disregard outliers, it can be helpful to look at the data without them so that we can get a sense of the distribution of the majority of the population. So if we take away that outlier individual, we have this, which shows a pretty normal distribution. Here we can see that the double extractions, like we saw with the histogram, have a large range and a slight right skew, while the singles distribute fairly close to normally. As far as statistics go, these numbers remain steady. The p-value dropped and remained significant, and the percent increase was only dropped by 4%. It's only a 171% increase as opposed to 176. So there you have it, a mostly complete rundown of everything we've done over the past 2 years. In orange, I have the issues that we're currently working on. As I said in the beginning, this is definitely an ongoing process. Finally, I have so many people and organizations to thank for making this possible, including the LMU Summer Undergraduate Research Program and Cadner Pitts Award, the Santa Monica Bay Audubon Society, Wilson Ornithological Society BERT Mentoring Grant, the NENHC Committee, and the Shoals Marine Laboratory Summer Internship Program. I would also like to thank Brielle Mishner in this lovely picture right here for conducting all the fieldwork for this study. Our permits are listed down here. And thank you so much for listening. I'm happy to answer any questions for you, so please feel free to email me or my advisor. You can also follow her on Twitter. And thank you again for your time.